To Biochem 1, we're going to start with the cell cycle, but first we're going to talk a little bit about the experiments. What you need to know, not in too much detail, but the general gist of it is the Miller-Urey experiment. That simulates Earth primordial conditions and basically proved that in those conditions we can create protein and RNA, the basis of our genetic evolution. Griffith's experiment is when they found that a dead pathogen can transfer its virulence to a live pathogen, and that can infect other organisms, such as this poor little mouse here. All right, let's begin with the cell cycle. So how I learned this was that G0 is our maturation phase. It's basically, if you can think of cells as a person or people, G0 is single life, right? You're just maturing, you're growing. The notable exceptions are neurons and skeletal muscle. Neurons stay in G0 forever because they don't divide. They don't go through mitosis at all. Skeletal muscle, they just have a longer life, so it's not as frequent as, an erythrocyte or a leukocyte, because those undergo mitosis fairly often. So G1 is starting like the duplication process, and it's kind of like when you're saving up for a family, right? And the exceptions are stem cells, because those cells constantly divide, so they're perpetually in G1, or they never enter G1, pardon me. S phase is DNA replication and histone synthesis. Histone is a protein that is involved in replication. It assists in basically like loosening up the chromatin, so it can be processed and read, but we'll go through that in another lecture. S phase is how I think of it as like stabilization, right? So you're finally saving up for a family, but now we need a stable career, we need a stable like home life. G2 is basically the last minute prep for the kid, and then mitosis is when your family expands. So this is all controlled by something called CYCLIN-dependent kinase. It phosphorylates these cyclin proteins, which are signal proteins, and it signals which phase to go through. And as you can see here, this basically, like the peaks tell you when things are starting to transform from one phase to another. So the G1S cyclin peaks when G1 is about to go into S phase, and M cyclin peaks when G2 passes into M phase. But G1 is the notable exception. How come this cyclin concentration kind of spans the entire expression cycle? Well, first we got to understand that our cells only divide in the presence of mitogens, which are protein signal molecules, and the mitogens signal the G1 cyclins, which trigger the cells to enter the start phase in G1. G1 regulates start phase by turning on the expressions of the G1S and the S cyclins, so it lasts during those two concentration peaks, as you can see here. These two coincide with the peak of G1, see? And once the concentration of those two decrease, so does G1, because G1 basically encourages the cell to start dividing. But once they enter mitosis, we don't need that encouragement anymore. We have already committed to mitosis in the prophase phase, and now the signal doesn't need to be there anymore, and it will degrade. So the cyclin-dependent kinases bind to certain cyclins in order to regulate the cycle. This is particularly important because once a cell is either damaged or too small, it won't continue. As you can see here, G1 checkpoint, that is basically, is the cell big enough? Does it have enough nutrients, and does it have enough organelles in order to duplicate. And cyclin D will bind to cyclin dependent kinase 4. Once those two binds, that's basically the go ahead to go into the S phase. There is a checkpoint in S phase that checks for DNA damage, replication, and CDK2 will bind to cyclin A, and that's the go ahead to go into G2. The G2's checkpoint is if size is big enough, if the DNA replication is good, and once cyclin B binds to CDK1, it will go from M into G1. So the notable exceptions to this is the RB1 and E2F. You don't need to know too much about that right now, but basically these are like oncogenes. Those are expressed if the DNA is damaged. If those aren't regulated, despite the damaged DNA, the cell will continue to proliferate. Mitosis versus meiosis, we should all know this by now. The most important thing is that non-disjunction happens, right? And those are heritable defects. Okay, let's see. This is the centrosome cycle. Most notable is that it starts in G1 and it completes in G2. Right. Prophase. So prophase does actually have 5 separate phases, as you can see here. What's different is that the pachytene, they have something called the synapnemo complex, and that's when the two chromosomes, they are bound together completely, and then it dissociates in diplotin. And dikinesis is when the nuclear membrane starts breaking apart, and that signals the end of prophase 1. I think I've mentioned it before, but prophase is the commitment step. If there's no protein to signal that prophase has started, the cell will not continue to M phase. All right, the kinetochores, those are pretty important. They are little proteins that are expressed. In meiosis 1, it's expressed on only one side, right? So when the homologous chromosomes are pulled apart, they're still paired. That's why it's only on one side. In meiosis 2 and in mitosis 1, they're expressed bilaterally, so the chromatids can be pulled apart. Another thing to know is that there is a cohesion protein on the centromere, and those compact the centrosome, keeping it, compacted in the nucleus, and that is for storage purposes. But once the spindle fibers from the centrioles attach, it reduces the amount of cohesion. As you can see, kinetochore increases, cohesion decreases. Why do we want that? Cohesion holds the chromosomes together, so we want it to decrease so it can be pulled apart. Okay, part 2. Part 2 is the cell membrane. We all know this. The lipid bilayer, glycolipid, and the glycoproteins. All right, so these are the 5 most common phospholipids, but what's more important is why these two are outside of the outermost layer, right? So memorization doesn't do you any favors in biochem. Understanding why these proteins and these various other biomolecules interact the way it is more important. So, okay, enough of that tangent. Sphingomyelin is on the outside. Why is that? So remember that, myelin is what insulates and protects the axons. So sphingomyelin is the phospholipid that will protect and lubricate the phospholipid bilayer. Phosphatidylcholine is it interacts with acetylcholine, and as you know, acetylcholine is a neurotransmitter, but it's a neurotransmitter that is a peptide, so that doesn't cross the lipid bilayer. So this has to be on the outside so it can interact with acetylcholine. Cholesterol. Cholesterol is the most abundant molecule besides the phospholipids in the entire membrane because it helps with fluidity. It's pretty rigid, as you can see, the hydrocarbon rings, and it prevents the lipids to basically bind together. It gives a little bit of gap so these two can still wiggle around. That increases fluidity. And it prevents crystallization when temperatures go down because, like I said, it doesn't allow the phospholipid heads to bind to each other. Right, membrane proteins are really important. They have a variety of functions: enzymes, hormones, signaling, this, that, and the other. Membrane carbohydrates are also very important. What's new is that a glycocalyx will protrude on the outside. So calyx is, I want to say Greek, don't quote me on it, but it means spear. So the portion, the fatty portion, will protrude outside, and that has a function in cell recognition. Right, more fluidity. So the fluid mosaic model, we all know that. Short chain fatty acids have a lower melting point, so that increases the fluidity. Unsaturated fatty acid increase fluidity because of the kinks. So remember that the unsaturated fatty acids, the double bond, like they're usually trans, right? And they don't let it stack as nicely and as neatly as a saturated fatty acid. Selective permeability. So we have to remember that the phospholipid bilayer is nonpolar, right? So the small nonpolar molecules are soluble and they can cross easily. But because they are, uncharged, even tiny things like protons, charged molecules, they can't diffuse through it. So they have to go through something called the transmembrane protein. And transmembrane proteins can be ion channels, active transport channels, protein ligand channels, things like that. But they allow the molecules to cross the membrane without interacting with the membrane itself. Transport is pretty important. We should all know that there's the different types, active versus passive. Everything is based on the gradient. And when it comes to diffusion, facilitated diffusion is also a passive transport. The difference is that it uses something else to assist in crossing the membrane. For example, an ion may not be able to cross, but a glycoprotein may. So the glycoprotein and the ion will bind, allowing things to cross. Usually in one direction, that's why it's a uniport. So it's either in or out of the membrane. Active transport, you should know about the cotransport, which is also known as a symport, and antiport. A symport will carry both the, let's say, like enzyme and the coenzyme in against the concentration gradient. That's the difference between a facilitated diffusion and symports. Well, besides, symports being an active transport, but it is because it is going against the concentration gradient. It's going towards a higher concentration gradient versus something like a uniport, which will go passively from high to low. Antiports will exchange it, so they will go, like sometimes it can follow the concentration gradient, but sometimes it doesn't. That's why it also uses ATP in order to do so. So as you can see right here. Osmosis will cross through aquaporins much faster because it's a facilitated diffusion, and it is a small molecule in relation to other things, such as like glucose, right? It's small, and even though it's polar, it can cross the membrane, but it is quite slow. That's why it usually goes through the membrane via the aquaporins. So we should study this chart. It basically is very, very helpful to know. Don't memorize things. That's my final advice. You should really be studying why these things happen. Like none of biochemistry happens because of like spontaneous reasons. There is a rhyme and a reason to the way these molecules interact with each other. Okay, that's it for now. Thank you for your attention, and I hope to see you in the next video.